DNA replication is the process where a cell copies its DNA before cell division. This ensures that each new cell receives an identical set of genetic information, DNA.
First, several enzymes work together to unzip and copy the DNA. Helicase starts the process by breaking down the hydrogen bonds between the base pairs, separating and unzipping the two DNA strands and forming a replication fork. As the DNA is unwinding, topoisomerase works ahead of the helicase to relieve the tension and prevent the DNA from becoming overwound or supercoiled.
Next, primase adds short RNA primers to the exposed DNA strands. These primers are necessary because DNA polymerase can't start building a new DNA strand on its own. DNA polymerase then adds complementary DNA nucleotides to the primer, synthesizing the new strands in the 5-3 direction. On one strand, the leading strand, this process is continuous, while on other strands, the lagging strand, it happens in pieces.
Once the DNA has been synthesized, the DNA ligase finishes the job by sealing the gaps in the sugar phosphate backbone. It connects the fragments of the DNA on the lagging strand, forming a complete and continuous strand.
DNA replication is described as semi-conservative. This means that each new DNA molecule contains the original strand and one newly synthesized strand. This preserves the original genetic info while still allowing for accurate copying.
The two strands of DNA are called the leading strand and the lagging strand. The leading strand is synthesized continuously towards the replication fork because it runs in the correct direction of DNA polymerase. The lagging strand is synthesized continuously away from the replication fork in short segments. The short segments are called Okazaki fragments and are later glued together by DNA ligase to make one continuous strand.
The difference in the two strands occur because DNA has directionality. Each strand has a 5' and a 3' and DNA polymerase can only add nucleotides to the 3' of a growing strand. As a result, DNA can only be synthesized in a 5-3 direction, even though the two template strands run in opposite directions. This means that the lagging strand, which is 3-5, has some extra work to do because it must be synthesized directionally in Okazaki fragments that need new RNA primer, while the leading strand can be synthesized continuously toward the replication fork.